So what we're going to be talking about today is using autosomal DNA that has been isolated from rootless hair. And it's always been...
I waited for you to start.
It has been dogma that when you're trying to get DNA from hair, that you needed to have the root. And if you didn't have the root, then the only DNA that was going to be in the hair follicle was mitochondrial DNA. So this is a very new concept, and we've really put it to good use. So just really quickly, a little bit on my background. I have a PhD in biology from University of California, San Diego, and a law degree from the University of Texas Law School in Austin. I'm a retired patent attorney. I actually specialized in doing biotech inventions. I lived in Palo Alto, and of course there were lots of little startups in the Bay Area. So lots of interesting work that we did. One of my big clients was a company called Calgene. They were up in Davis, California. And one of the patents that I worked on for Calgene was the flavor saver tomato. And this was a tomato that had been genetically engineered so that it didn't ripen. We were blocking the enzyme that allowed the skin to basically to rot. And so you could then pick the tomatoes ripe. So in their wisdom, Calgene hired somebody for a new company they were setting up called CalFresh that was going to be marketing these tomatoes. And the person was from Chicago, so they decided to put CalFresh in Chicago. So their first year, they had contracts with US farmers to grow these tomatoes. Somewhere along the line, the farmers realized that these were genetically modified, and they decided they weren't going to grow the tomatoes. So we then had to go another year, and they made contracts with farmers in Mexico. So the farmers grew the tomatoes got picked, loaded into the trucks to go to Chicago. But what everybody forgot was that they were ripe tomatoes, they weren't green tomatoes. And so when the trucks got to Chicago, there was so much tomato paste running out of the back of the trucks. So that was the flavor saver tomato. In terms of working with investigative genetic genealogy, the first case I worked on is one I'm going to mention a little bit this morning. It was the Lisa Jensen case. And I started actually working on that in 2015. We solved that case in 2016. 2018, I worked on the Golden State Killer case, and I did talk about that particular case last time I presented here at Dent. And then since then, I've assisted in solving about another 50 cases. I'm told that I've probably solved more cases than most detectives. So for full disclosure, I am the Director of Investigative Genetic Genealogy at Gene by Gene, which is the parent company of Family Tree DNA. So what I'm going to cover today is the impact, actually, of solving the Golden State Killer case. It's really been significant. I'm also going to talk a little bit about what exactly investigative genetic genealogy is, and then I'm going to talk about 3 cases that I've been working on using the autosomal DNA from the hair shaft. The cases are, we've solved 3 of the victims of Bear Brook Park. For those of you who listen to podcasts on true crime, there have been several podcasts on those cases. And then who murdered a little girl called Darylyn Johnson in Nampa, Idaho. And then finally, who was Precious Jane Doe from Snohomish County in Washington. So just to put things in perspective, the structure of DNA was first published back in 1953 by Watson and Crick in their seminal paper in Nature on the structure of DNA. And this, of course, had a profound effect on the biological sciences. This was just such a key finding. 65 years later, ironically, on April 25th, 2018, the Sacramento County DA's office announced that the day before they had arrested Joseph James D'Angelo on suspicion that he was the Golden State Killer. And on that day, the world changed for criminals who have left their DNA at a crime scene. This was dubbed as being the DNA hit of the year in 2018. And it has really profoundly changed, as I said, how cases are being solved once we have evidence which has got DNA attached to it. Joseph D'Angelo was sentenced this last August to 11 consecutive life terms, plus another 15 life terms plus 8 years to be served concurrently with the other life sentences. This is all without possibility of parole. So these are the people who were on the team that worked on this case. There was Paul Holes, who actually had been the detective working on that case. Stephen Kramer and Melissa Paraso from the Los Angeles field office of the FBI. Monica Kozowski and Kurt Campbell from the Sacramento County DA's office, and then myself. These are some numbers that are associated with the traditional investigation into who the Golden State Killer was. So it was a 43-year-old cold case when we started working on it. I'm not going to remember what all the numbers were. I forgot to bring my little cheat sheet. But there were something like 650 agents and detectives who had worked on this. There had been 300 people that they had actually done surreptitious DNA testing on. They'd put 8,000 people under surveillance. And all of this had cost $10 million. For the investigative genetic genealogy portion of this, we spent $217 on a DNA kit for doing a consensual sample. We took 63 days from when we first worked on this case to when we actually solved it. There were 6 of us working on it, and we came up with 1 suspect. So obviously this is an incredibly efficient way of doing research into identifying suspects. There's well, I won't read the whole slide, but basically you're saving money, you're saving shoe leather, you don't have so much manpower invested in doing this. Yes, it is time intensive to do the genetic genealogy if you don't have very close matches, but it's still a much more efficient way of identifying suspects and unidentified human remains than traditional methods. So this is in just a little group picture of us. This is on the day of the sentencing. From left to right is Monica Kozowski, looking very FBI-ish in the sunglasses, as Steve Kramer, Kurt Campbell, and then sort of going incognito with a beard is Paul Holes, and then myself. And then Melissa Paraso had been reassigned to the Washington office, so she wasn't there. So Paul Holes had actually worked on this case for 22 years. And as I said, we actually identified him in 63 days from upload to GEDmatch. And I'm going to talk a little bit more about GEDmatch in a few minutes. There have been now over 300 additional cases that have been solved since GSK was arrested. And I myself and my team, we've solved about another 50 cases. So exactly what is investigative genetic genealogy? So genetic genealogy itself is basically doing regular family history type research, but adding in the component of using DNA. And this can then be used to either refute, confirm, or maybe augment the research that you've done using traditional methods. So traditional methods being using things like census records, newspaper articles, voting records, whatever's out there that is going to allow you to follow where your ancestors were. It can also, of course, be used for unknown parentage searches, so people who are adoptees looking for their biological relatives, whether it's parents, grandparents, people who were donor conceived, which is, a little more controversial because, of course, people who were sperm donors or egg donors were guaranteed anonymity at the time of their donation. Foundlings, one of the cases I worked on was David Volk, who had been abandoned as an infant, a newborn infant, in a McDonald's ladies' room. And then victims of crime, of course the case I worked on was Lisa Jensen, who had been abducted when she was about 6 months old. So there's a difference in the type of samples that you actually use from those of you who have done DNA testing, if you tested Ancestry or at 23andMe, you're going to be spitting in a tube. And if you tested at MyHeritage or Family Tree DNA, you're going to do a little buccal swab. And then you're identifying genetic relatives in the databases of the companies where you have tested. And also, of course, you can do an upload to GEDmatch. You're doing exactly the same thing you should do for family history research. You're doing, you're building trees, you're using census records, newspaper articles, same stuff. It's just that the source of the DNA has changed. So forensic samples then are typically biological fluids, like blood, semen, saliva. You can also, of course, use hair, and I'm going to talk today about rootless hair. And if you're doing unidentified remains, of course, you can also use things like bones and teeth. For doing forensic samples, there are only two databases that you can actually use, one of them being GEDmatch and the other one being Family Tree DNA. There's also a slight difference in some of the results that you get and also the cost. So when you're doing direct-to-consumer testing, you're using nice, clean, spit or buccal swabs, and so that testing can be done by robots. And if you watch, there's a recent YouTube that was done by Family Tree DNA, and you could see that everything basically from when the tubes with the buccal swab in them arrive in Houston, they're immediately put into this robotic system which does everything from the beginning to the end. With the forensic samples, of course, you can't do that. You've got all kinds of different samples, and they can be more difficult to extract. You may need to do some slightly different testing. If the DNA is damaged, you probably can't do the traditional, what we call SNP array, which is what Ancestry and the other companies use. You may have to do what's called whole genome sequencing, where basically you take the DNA, if it's not already in pieces, then you're going to put it into small pieces, put it all back together again, and then you're going to have to do genomics on it to pull out the equivalent of the SNP array that people have done at one of the other companies. Obviously, this is all a lot more expensive to do than the robotic stuff you can do with the direct-to-consumer tests. So when do you want to do this? Well, you obviously have to have DNA evidence. You preferably want to have a CODIS profile, and of course you're going to be doing this when you have got no hits in CODIS. And CODIS is the database that is run by the FBI. It has in it samples that are from people who've been arrested in various crimes, people who are suspects in crimes, and also crime scene DNA. And you're also typically going to be doing this because of the cost when you've exhausted all other areas of research.
CODIS database has got DNA sequence information on the suspect?
Excuse me?
Does the CODIS database have the DNA sequence from a sample, or how do you get that information?
Yeah, that's a good question. So the CODIS database is actually a little different from what we're doing. It's still autosomal DNA, but it's what are called short tandem repeats, STRs. And there are 20 of those that are used in comparing the different people who are in the database. We're using the autosomal DNA that is from the 23 pairs of chromosomes. So we're looking at 700 to 800,000 SNPs, so a very different type of testing.
So my question is, how does a criminal get his DNA into the database? What physically is happening?
He's been arrested, and so there's been a search warrant, and so he's had a sample taken, and that's what's put in CODIS.
And what kind of sample is being taken?
They typically do a buccal swab. Okay, so there are 3 DNA tests then typically. So for those of you who've done testing, there's Y-DNA, there's mitochondrial DNA, and there's autosomal DNA. So Y-DNA, of course, is looking at your direct paternal line, who's your daddy. And that's my daddy, and back up my paternal line. And then, so the Y-DNA is, basically you can go back probably about 25 generations. And the DNA gets passed from father to son, to his son, to his son, to his son, virtually unchanged through all of those generations. The mitochondrial DNA is doing sort of the same thing on the maternal line. There's a slightly different inheritance pattern, but it is still passed on virtually unchanged from one generation to the next. What is different is that although a mother passes her mitochondrial DNA onto all of her children, we're going to have the, oh no, this is quarter off. She passes it on to all of her children, but only the female children typically pass it on to the next generation. There are some occasional reports of the father actually passing on his mitochondrial DNA, but that's extremely rare. You're again looking at, you can go back many, many generations, and so again, very stable. So the mitochondria, for those who are not familiar with it, they're these little oblong things that are in the cytoplasm of the cell. Depending on the cell, there may be just a handful of mitochondria, or if it's a cell that needs a lot of energy, the mitochondria are actually the power pack for the cell. For those of you who remember biochemistry, this is where you make ATP. And so if it's a muscle cell, you're going to have lots and lots and lots of mitochondria in those cells. It's believed to be bacterial in origin, and the DNA is that little circular piece. It's a double-stranded circular DNA. So this is then just a diagram showing the mitochondria coming from the mother, not from the father. But the DNA that we're really interested in for doing investigative genetic genealogy is autosomal DNA. And this is different from the CODIS DNA. This is now, we're looking at SNPs. So unlike the Y-DNA and the mitochondrial DNA, which get passed virtually unchanged from one generation to the next, the autosomal DNA goes through a mixing and matching process at each generation. So although you inherit, you and your siblings will have each inherited half of your DNA from each of your parents, you've each actually gotten a different random mix. So if you've got blonde hair and blue eyes, and your brother has dark hair and green eyes, that's why. You got totally different random mixes of your parents' DNA. So this very complicated looking diagram is just a summary of what I've just said. So is there a pointer in here? I didn't ask.
It won't display on the screen.
It won't? Okay. All right, so I always avoid doing this because I'm not very tall, but I can actually do it here. All right, so let's just really quickly then go over what I've just said. So we've got Sonny Boy down here. He's got a Y chromosome, which is shown here in the blue, and we're showing it's coming down from the paternal line. We've got the mitochondrial DNA over here, so Sonny Boy has gotten his mitochondrial DNA from his mom, and again, it's come down the maternal line. And then we've got a pair of autosomes, and this can be, you've got 22 pairs, and so this can be any of those pairs. So let's say this is a pair of chromosome 3. You've got one copy from the paternal side, one copy from the maternal side. And what has happened at each generation, as you can see from all the hashing and the colors, is you've gotten this mixing and matching that's gone on. It's called recombination. So for doing investigative genetic genealogy, what we're doing is, let's say Sonny Boy here he's done his matching. He's got a match with a second cousin. Second cousins share a set of great-grandparents. So if we were to look, say the matching is on this yellow piece of DNA here. So that they're second cousins, we know we want one of the great-grandparents up here. So we can trace up here, and we can see that it came from this set of great-grandparents up here. So we know then that the second cousin and Sonny Boy have got this shared great-grandparent, and they're both descendants of that shared great-grandparent. Let's now add a suspect to the mix. He's got his DNA in the system. He has a second cousin match with Sonny Boy and the other second cousin, and they're all matching on the same segment of DNA. What do we know about the suspect? We know the suspect has got the set of great-grandparents in his ancestry. What else do we know? Our suspect must be a descendant of that couple. So what we do is we go ahead and we now look for all the descendants of that couple. Our suspect is amongst those descendants. What we have done is we'll have done a little profile on the person we're looking for. So for example, for GSK, we know that the first reported rapes that we were aware of were in 1976. So that means he was probably born in 1956 or earlier. We know he's in Northern California. We know some other things from eyewitness reports, about how tall he is, what his his hair color is, and so on. So once you've gotten all your descendants of your couple, you then just go through them. Do any of those people match the profile that you have? So that's basically, in a nutshell, what you're doing. So this is then the little protocol. We start off with our DNA sample we've extracted the DNA from. We've gotten our SNP array. This is showing what a SNP is. It's a change in one of the nucleotides. And as I said, we're looking at 700,000 to 800,000 of those. We can then download that information. It's downloaded as either a text file or a CSV file. You then upload it to one of these two companies, GEDmatch or Family Tree DNA. And that's then going to give you a list of people with their email address, the amount of matching DNA, likely relationship that you can then look at. Then from there, you do what I just said. We're building family trees. We're looking for our common ancestor, identifying all the descendants, comparing that to the profile, and then we get a little list of candidates for who our suspect is. And then if we're down to one or two, we can then go out and get some surreptitious DNA, see if it matches the crime scene DNA. Voila. Okay, so when you're doing this, what you really want, obviously, is a second cousin or better, because you've then got a relatively close, most recent common ancestor. It's a doable number of people to identify the descendants at that point. If you start going further back, it becomes more difficult.
Because they had matches, their cousins, Sonny Boy to the cousin had similar matches, right? So then do they both become suspects?
No, they're what you're using to build the tree. That's a very good question. And then you've got your unknown, which is the suspect.
I see.
And so then you're going to be then seeing, okay, amongst all those descendants, who do we have that's fitting the profile?
But if you got a match, then you actually already have a suspect, and you don't need genealogy, right? That's the...
No, because you have the match is just, it's going to be at second cousin or third cousin. And so you then have to identify who that second or third cousin is. Yeah. So you still have to do with the build down in order to identify who the person is. But you can use the fact that they're a second cousin or a third cousin to narrow down your pool. Okay, so this is then going through looking at how the second cousins are related in a slightly different format. You've got the match, and then the second cousin, and then the shared great-grandparents that you're going to be building down from. So how likely is it that you're going to get some second cousins or better? Well, so these are sort of ranges of how many relatives people have. Most people don't have that many first cousins, or second cousins for that matter, but they do have lots and lots of third cousins and fourth cousins. So those of you who've done DNA testing, you'll have seen you've got this long, pages and pages and pages of matches, sometimes 3,000 and 4,000, but they're mostly going to be the more distant ones. So what then are the odds of you getting those matches? Well, if you look at the US population, it's about 327 million. To get a second cousin, if you've got 1% of the population, and you've got a 58% chance of getting a second cousin. If you've got a bigger sample, then you've got pretty good chances that you'll get that. But the databases that we're working with don't have that many people in them. So the databases we're working with are these two down here. The red one, which is Family Tree DNA, and the gray one, which is GEDmatch. So Family Tree DNA has maybe got 2 million people in it, And GEDmatch, even though it's probably got about 2 million people in it, not everybody has what's called opted in to letting people who, to letting there be matching with a law enforcement kit.
And yours is the family tree DNA and MyHeritage? Those are your two?
No, so it's unfortunately it's these two. MyHeritage is up here. It's not one that accepts uploads. Yeah, what we'd really like to have is is all those folks in Ancestry and 23andMe, but they don't want us in there. Okay, and this is just showing what the definition is of a centimorgan, which is the unit of measurement for how much matching DNA there is. So these are the main direct-to-consumer companies in the US. And then this is GEDmatch. So GEDmatch accepts uploads of a DNA profile from basically any place, including the forensic companies. And what's important is that it's a voluntary upload. There's no DNA that's transferred. All you're doing is uploading that CSV or text file. A lot of people think that they're uploading their DNA. They're not. Their DNA is staying wherever they tested. GEDmatch itself does not do any testing, and it is a public database. Okay, so what have I done lately that's denty? So I'm going to really quickly go through some of the original stuff and then get to what's new and denty. So I originally started off, as I mentioned, with Lisa Jensen, and her picture is deliberately distorted because apparently her children look, or at least she's got 2 little girls who look a lot like her at that age. So back in 2015, Deputy Peter Headley in the San Bernardino Sheriff's Department Crimes Against Children Detail contacted DNA Adoption, which is where I was volunteering as a search angel, to find out if the technique that we were teaching to adoptees could be used to identify somebody like Lisa who'd been abducted as an infant. In July of 2016, we identified who she was. Her mother was a woman called Denise Bodan from Manchester, New Hampshire. And she had been missing, along with her baby Dawn, who presumably is Lisa, since 1981, and they'd never been reported missing. So I had also, of course, identified some potential people for who Lisa's father was. But it turned out that Denise's father, so this is now Lisa's grandfather, is still alive. So we asked him, so who is Lisa's father? Bob Evans, he says. Well, Bob Evans wasn't one of the names I'd come up with. So on a hunch, the deputy sent a picture of the guy who had abducted Lisa to the grandfather. Yes, that's Bob Evans. So it turned out that the person who was the abductor had actually been her mother's live-in boyfriend. He had a whole bunch of aliases. When he was with Lisa, he was using the name Gordon Jensen. And then he was arrested in California for the murder of his common-law wife, Eun Son-jun. He was using the alias Curtis Kimball. So there were some similarities between the MO for how Eun Son-jun was killed and the murder of 4 people in a place called Allenstown, New Hampshire, which is about 20 minutes from Manchester, New Hampshire, where Denise and her boyfriend were living. So this is, it's called Bearbrook Park, or these were called the Allenstown 4. So there were an adult female and 3 little girls. They'd all been murdered by blunt force trauma. The adult female and the eldest child had been dismembered, wrapped in plastic, tied with electrical tape, and then stuffed into that first steel drum up there on top left. This is very similar to Eun Son-jun. She had been killed by blunt force trauma. She'd been dismembered, wrapped in plastic, tied with electrical tape. In her case, she'd been buried under 200 pounds of kitty litter in the basement of her home. In the second barrel on the bottom there, excuse me, was 2 little girls. The other 2 little girls were found in 2000. It does punctuate at the right times. So at some point, the New Hampshire State Police connected all the dots. And they announced in January of 2017 that, in fact, Bob Evans was the murderer of these 4 women, or these 4 females. And then there was an article that was published in Forensic Magazine talking about how, basically, because I had identified who Lisa's mother was, they'd then been able to solve who had done these terrible murders. Of course, we still didn't know who he was. But meantime, the question was, is the adult female the missing Denise Boudin? And so DNA was looked at on all of these. The adult female was not Denise Boudin, but the little girl on the far right with the dark hair, she was actually the killer's daughter. So then, of course, the question is, well, where's her mother? Is she another victim? Who are the Allenstown 4? And who is Bob Evans? So we tried many, many times to try and get autosomal DNA from the remains of the Allenstown 4. We tried with different companies, and basically what we would end up with, the bodies were so contaminated because they'd been out there for so long that we were just getting basically bacterial DNA. So we would get 98% bacterial DNA, maybe 2% human. Theoretically, you can amplify the human that we were getting, but it would have been extraordinarily expensive. So basically, this was a dead end. In March of 2017, I had to have open heart surgery, double bypass, mitral valve repair, and I'm recuperating at home, bored, surfing on the internet, and I come across this newspaper article in one of the San Francisco newspapers about the identification of a little girl called Miranda Eve. Of course, I'm fascinated, particularly when I find out that what was used to identify her was rootless hair. I was so excited. I mean, this was because we had hair on all of our Allenstown people. And so it turns out that Dr. Ed Green at the University of California at Santa Cruz, which is basically right up the street from where I live in Monterey, had worked on doing this. Of course, what they were doing, so this is the house that's being renovated in San Francisco. It turns out that back in the day, San Francisco had moved all of their burial grounds out of the city to Colma, which is in South San Francisco. And they apparently had missed a few coffins, and this was one of them. So it had this little girl in it. And so what Dr. Green had done is he'd taken some strands of hair, and he had isolated autosomal DNA out of these hair strands. Well, he of course was used to working on old things. He has a paleontology lab. They work on dinosaurs and bison and other old things. And so they had a technique for getting stuff out. Of course, they weren't doing what we typically do. They looked for somebody who was a relative. So they had aerial photographs of the area where this house was, and they were able to determine that actually where it was built was on what had been the site of the Oddfellows Cemetery. And they could identify probably which grave it was that this coffin had come from. And so they had been able to identify this fellow, Peter Cook, who lived across the bay in Marin. And so basically all they were doing was comparing DNA. And were able to show, in fact, he was probably the little girl's great-great-nephew. So they were able to identify who that was. So as soon as I am somewhat recovered from my surgery, I'm on the road up towards Santa Cruz to meet with Dr. Green to find out if he's willing to work on forensic samples. So we sort of met halfway. There's a nice restaurant called the Whole Enchilada in Moss Landing, and so we had lunch there, talked about doing forensic samples. And he was kind of excited about doing that. And so, In the meantime, we've, working with another lady in my group, we'd actually identified who Bob Evans was. Turns out he was a guy called Terry Pater Rasmussen from Colorado. And he had actually died in prison in California in 2010, which is actually how I was able to identify him, because California actually autopsies all of its deceased prisoners. And so I was actually able to get a blood card from him and figure out who he was. So from May then of 2017, when I got together with Dr. Green at the whole enchilada, through to November of 2017, we worked on trying to make this process work for forensic samples. And so we kept uploading files. And of course, what I had some really good internal controls. So but let's take a look then at why people didn't know that the DNA was there. It turns out that the fragments of DNA in the hair follicle are really small. Their average size was about 45 base pairs. And the traditional type of technology, PCR technology, polymerase chain reaction, that is used for identifying DNA uses these two tags here, P5 and P7. The fragment of DNA that goes inside that has to be a minimum of 150 base pairs through to around 600 base pairs. So these fragments were too small to be detected. Did you have a question?
Yeah, so y'all still had the deceased Allens, the Allen people, right?
Mm-hmm.
So, and you couldn't use the hair follicle line. So why couldn't you use the hair follicle? I know we're talking about like not having the follicle and finding it that way, but did you have the hair follicle on the?
We had the hair strands. We didn't have, there were no roots.
Oh, I see.
So that's exactly what we worked on, was the hair strand.
Got it, okay.
But yeah, because I mean, they'd been out there for,
And their follicle, I guess, died?
yeah, it was, yeah. Or they got contaminated. So yeah, I mean, they were in pretty sad shape, those bodies. So so Dr. Green developed this protocol. So basically you just extract the DNA that we now know is there from the hair shaft and purify it. You then just develop some sequencing libraries, and then actually do whole genome sequencing on this, which is basically putting all of the DNA, all those little tiny pieces back together again. And then you do sequence analysis and genotyping. And then what you then need, of course, to do is we need a file that we can upload to GEBmatch and FamiTrueDNA. So for some of the sites, we could actually call what the nucleotide is that should be there. For some of them, I used what's called imputation, which is basically if you know what, say, 2 other sites, what those are, you can guess with a fair degree of reliability what the remaining sites should be. And so then you can come up with what's called a SNP profile. So it sort of looks like what you would get if you tested at Ancestry. The other cool thing about hair is that you can actually sterilize it. Because, of course, contamination had been one of our big problems with using bones or tissue. You can actually sterilize it with bleach. And, of course, one hair comes from one person. It's also a single source. And for a lot of forensic samples, particularly like rape kits, quite often they're contaminated with the victim's DNA. And so you then have to try and sort out which, get the male fraction, basically. But with a hair, one person. So what we had that we could work with to try and make sure that this was really working was we had 2 sets of parents and children. So we were fairly sure that for the Allenstown folks, we knew that the 2 children were mitochondrially related to the adult female. We guessed she was probably mom. So we had the adult female, then we had the 2 children, and then of course we had Rasmussen and his daughter. So we could use these, and so we would do a file, would upload it, were we getting the right kind of matching? So I think for the first 4 or 5 tries, we weren't getting the proper parent-child matching. And so we just kept going, tweaking the algorithm that we were using to come up with our SNP profile until we got the right amount of matching. So basically what you're trying to do is that for a parent-child match, you should be getting around 3,500 centimorgans of matching. For siblings, so if those 2 little girls were for full siblings, it'd be around 2600. If they were half siblings, around 1750. And so we would go through and look at this. So what we ended up getting then, when we finally got the algorithm working properly, we then determined that the two little girls were in fact half siblings. And from there, we were actually able then to upload the files to GEDmatch and do our typical thing of figuring out, building the trees, doing our most recent common ancestor, and then identifying who they were. So the adult female turned out to be a woman called Marlies Honeychurch. The eldest child was Marie Vaughan, and the other child was Sarah McWaters. We're still working on identifying Rasmussen's daughter. So this was really kind of cool. And so the question was, can we use this on some other cases that we have? So this was a case out of Nampa, Idaho, a little girl called Darylyn Johnson, a horrible rape homicide of this little girl. This was actually a case I worked on with the same FBI team that we worked on the GSK. So she had a 6-block walk to school. Somehow she managed to disappear in that little distance back in February 24th, 1982. And then several days later, she was found in a drainage ditch along the Snake River. Somebody had actually been convicted of her murder and had spent 18 years in jail. There was actually an Idaho Innocence Project. They actually used mitochondrial DNA to absolve him of the crime. So, and then since 2001, the case had gone cold. It was a single hair, male pubic hair, that was discovered on the little girl's body. And so we sent that to Dr. Green. He was able to get sufficient DNA out of that one pubic hair for us to identify a suspect.
When you only have one element of evidence from the perpetrator, and you're consuming it for making DNA that can lead to their capture and identification, is there pushback from law enforcement though from like consuming that one piece of evidence that they have in these efforts?
Yeah, and in fact, what had happened in this case is there had been other evidence, and there'd been a fire apparently, and this was actually all they had left was this one hair. And so actually in this case, there was not even any DNA in CODIS. And so it was kind of an interesting situation because if it had in fact, I don't think it's finalized yet, but if it had gone to trial, Dr. Green would actually have had to testify as to how we were getting to where we were. But we could at least take the DNA, get a search warrant for a DNA swab from the guy that we've identified and show that he matches with that hair. But it's not the usual route that you would take. So that's, it's a little bit tricky. So in this particular case, that hair gave us some incredible matches on GEDmatch. We had a 538 centimorgan match, which is probably a first cousin once removed, and then another one at 256. We almost didn't have to build a tree to identify who the perpetrator was. And so we came up with this guy called David Dalrymple, who actually had lived in Nampa, and he actually was already in prison for a similar crime. He was, however, going to be released on parole in 2023. So I don't think that's going to be happening anymore. And then we have Precious Jane Doe from Snohomish County in Washington. This is really kind of a sad story. She basically ran away from home. She was 17 years old. And from what we can figure out, she was murdered within less than a month of having run away from home. And unfortunately, now that I've worked on a number of these unidentified human remains cases, over and over and over again, we see these young girls who've run away from home, and in short order, they've run into somebody who didn't wish them well, and they've ended up deceased, which is very sad. So in this case, the guy who actually killed her had a few too many beers at the pub, and he bragged about having killed her. And so one of the people who overheard the conversation turned him in. And so he actually had, by the time we identified who she is, he had actually already served his sentence and has since died of cancer. But the cold case detectives actually met with him, and he claimed he never knew what her name was. What he did remember was that he'd wanted to have sex with her, she'd refused, and so he'd shot her in the head and strangled her with a bungee cord. So in her case, she had actually, where her body was kind of swampy, and so it had deteriorated fairly quickly. So again, we went through, trying multiple times to get DNA from another source, but finally we decided that that was not going to work. And it turns out, actually, for whatever reason, her hair was sitting in the evidence locker. So they actually didn't even have to exhume her to get the hair. So we sent the hair to Dr. Green, and he then developed a SNP profile, which we then uploaded to GEDmatch. And in her case, so she died in August of 1977. We figured that she was probably 16 to 19 years old. It turned out she was 17. And so we had a time period when we believed that she was born. We had this information on her admixture, which was basically Northern and Western European, that we could apply. So this then, I wanted to show this because it gives you an idea of when you're working with things like hair, it can be really kind of tricky. And so you have to really sort of look at what you have. So this then is, this is now looking at family tree DNA matches. So you, as you do with the other sites, you get a list of names and an email address, what the estimated relationship is, and how much matching DNA there is. So as you know, on these, we started out with little tiny fragments of DNA that we were trying to put back together again. So interestingly, the first one up there, the CA green, is actually the eldest child from Allenstown, another hair case. And so what you're seeing is there's a fair amount of matching DNA, 231 centimorgans. But if you look at the longest block, it's only 8 centimorgans. And typically, segments that are less than 10 centimorgans, they're probably not real. And so when you see something like that, it's probably noise. If we look, and again, if we look at the next person down, Nick V, we see 197 centimorgans, which is exciting, but the longest segment is only 8 centimorgans. And if you look at, I think this is Nick V over there, where the arrow is pointing, that one little tiny piece that it's pointing to in the middle of that, that's chromosome 3, that is the only segment that is bigger than 7 centimorgans. All the rest of that matching DNA is noise. So, and this is actually showing here then again, looking at just the 7 centimorgans and larger, that one little tiny blue spot is that segment from up there. So obviously we needed something better. And so we now then sorted on longest block, and then we come up with somebody who is a match. I think it's 119 centimorgans with a 71 centimorgan longest block. That's something you can work with. And so that then is a match that we're going to be working with. We also had, Dr. Green actually did another file because obviously there were some issues with the file we had. He did another file. This is now looking at GEDmatch. We've got a new, quote, big match at 85 centimorgans. And so we were building, and that was somebody who had tested at 23andMe and then uploaded to GEDmatch. And so we have built out a tree using our family tree DNA match, which when we adjusted it down to the 7 centimorgans was a big 86.4. We were now able to build out and get a most recent common ancestor, who was this Margaret Alt. So how to put all this together and try to sort something out with it? What we started doing is using lucid charts and scapels to try and visualize how these people are related to one another. So what we did here is, there's our FTDNA match, the one that we've recalculated at 86.4. We've built out the tree. We go up to our most recent common ancestor up there at Margaret Alt. And here's our new GEDmatch match at 85 centimorgans. And it turns out she actually had a sister on there at 71.1. So how do we make sense of this? So you do something like this. So you've got what we call the golden arches. We've got our most recent common ancestor here. We've got another one over there. And then we find that we have a connecting couple. And so we've got someone from this tree who is married to someone from that tree. And when you have that scenario, you know the person you're looking for is a descendant of that connecting couple. So we knew that this is where precious Jane Doe belonged, was here, because we know she's related to this person and she's related to that person. So we go to our tree, and what do we find? This couple has 3 little boys. Uh-oh. Jane Doe is missing. So one of the ladies in our group is really good at finding things in obscure places on the internet. She somehow managed to find a tree by a relative of these folks. And in that tree were our 3 little boys, and there was a little girl. It turned out that the parents had gotten divorced in Oregon. We were able to get a hold of their divorce records, and in it had that there was the 3 little boys and the little girl was planned to give her up for adoption. So we now had the name of the little girl, Elizabeth Marie Elder. But that's not her name, because this is the biological name. So it's kind of okay. So we approached the folks in Oregon. These people are dead. She's dead. They won't tell us what her adoptive name is. Privacy. And we're going, there's nobody whose privacy is being violated here. So finally, it took about 3-4 weeks for the detective to finally get the name of the adoptive parents. The adoptive father was actually still alive. And so in the meantime, we had then been looking for other relatives. We found that there was actually another child who had been born before this couple was married, and he had been given up for adoption. And we were able to find him. Because he was adopted, he was trying to find his birth relatives. He tested at Ancestry. We got him to upload to GEDmatch, and bingo, 1,813 centimorgans. He's a half-sibling. So we suddenly had some proof that we had the right person. And then, so we then also wanted, of course, to see that this was really making sense. I mean, we're working with these little tiny fragments of DNA, right? So, was this really legitimate? It certainly looked like it. But if we went back and looked at this, so our family tree DNA match had originally been 119 that we'd recalculated. Estimate would be somewhere between a second cousin once removed, third cousin once removed. In fact, was a third cousin once removed. Same thing with the GEDmatch person, same estimate, and was actually a third cousin. So this then was was precious Jane Doe. Her real name was Elizabeth Ann Roberts. Well, it was her adoptive name, and apparently she went by Lisa Roberts. So this is then just a quick summary of how we do this. We're building a speculative tree based on the matches that we have. We then add any matches that also have that same ancestry. We look for the most recent common ancestor, and then we find all the descendants. So for any of you who have done DNA testing, if you will please upload to GEDmatch and Family Tree DNA. If you haven't tested, if you will, and we'll do the same thing. That would be really cool. Encourage your friends and family to test. And that's it.
train like connected to the next one, like how it's kind of branches and spiders out.
Yeah, so there are several ways that you can figure out who the person is. That's one of them, is looking for the connection and then looking for the descendant of the connecting couple. The other thing is just to look at people within the tree and try to figure out who the person is from there. And that's actually what we did with GSK. So there's all kinds of different tricks that you try to do to try and figure out who it is. The most compelling, of course, is when you get the double arches, because you know if the person's related to people on both sides, that it's, it has to be somebody else that's in that same family.
So, how would we take ancestry DNA and upload it into GEDmatch?
So you can actually download, if you go into settings, there's a place where you can actually download, I think they call it download your raw DNA data file. So you download that file, and then you just register on the GEDmatch or on Family Tree DNA, and then you just upload that file and go from there.
Why is it that Ancestry and 23andMe don't want, I think it would be something
I honestly don't know. I mean, I think that they think that they would get backlash from their customers. But actually, when there have been polls done, something like 75% of people say they'd like to help law enforcement. And it's certainly what you see at Family Tree DNA and at GEDmatch, is that people overwhelmingly want to help. Yeah. So I think they should at least opt for the option.
maybe we could start out.
There you go. Yeah.
It should be an option. It should be forced. But I will say that, and I'm super happy to share my DNA. But that there are a lot of things that happen when you share your DNA. For example, you find out that your father isn't actually your father, right? Or you find out that your grandfather isn't actually your grandfather. So those stories that have circulated, that have happened already, like those are stories that are out there. I think that's what drives. I think that's the fear those companies have. Also, I do think that your grandfather doesn't want you to know, or your grandmother doesn't want you to know. Your grandmother doesn't. Yeah, I want to know.
I would want to know.
So one other thing is that I would say also is that it's kind of complicated. We just took a wonderful class to hear all about it, but I do think some people misunderstand that this is not giving you actual DNA. That you can't later be, you can't take this DNA and place it at a crime scene and then be convicted of a crime you didn't commit. And I did a study with one of my clients once talking about privacy of fingerprints and blood, and that was the number one concern. So I
What
wanted
would be the one thing that what it would actually put in?
it would exactly what it would do.
Yeah,
It would
someone
show.
taking
It would show that you didn't do it.
Right.
That's exactly right. That's exactly what it would show.
What would be the push
That's
then?
exactly what it would show.
But it might show that your cousin did it.
Yeah, which is a good thing, right? If your cousin is GSK, let's get him off the streets. Yeah.
Exactly.
He looks so. I mean, I don't want to say he looks young, but he looks so young. If he committed those crimes
He looks young.
40 years ago.
He was in really, really good physical shape. In fact, at the sentencing, Anne-Marie Schubert actually showed a little video of him doing, what do you call them, burpees in his cell. He's sitting there all pathetic in his chair.
In a wheelchair.
Yeah, but he was, that was just total sham.
He put himself
He's
in a
doing
wheelchair.
burpees in a wheelchair.
No.
It was totally a sham.
Oh my gosh. Yeah, because I was just thinking, so 40 years plus like 20 when he committed the crime.
Yeah, he was. So I think he's 70, maybe 75.
Okay. All right.
But when they had him under surveillance before they were, when they were going to do the surreptitious DNA, he was riding his motorcycle at 100 miles an hour on the freeway. There was nothing wrong with him. Yeah,
But he put himself in
Where
a
did,
wheelchair.
where did they catch him? Where was he living in the car?
he was actually living in Citrus Heights in Sacramento, in plain sight.
In plain, that's what's scary.
Did they ever tie him to any other murders that were like more contemporary? Like, did he continue to commit? I don't know.
It's a question that a lot of people are asking. Did he, because I think when the last murder was committed is sort of the very beginnings of some stuff with DNA, and it could be that that was why he stopped, or that he changed his MO. You know, instead of going after people in their homes and so on as he was, maybe he started going after prostitutes or whatever.
So I have a movie idea for you. Now you've solved this. I'm sure lots of people are sure about movie ideas. Now you've solved this. You go back and interview him and try to figure out what crimes he didn't admit to. Because there's just no way he committed all these crimes and then stopped 20 years ago.
He's not gonna give it up if he's doing burgers.
No, but but he has lifetime life. He has like multiple lifetime. He's not getting
What have you got to lose?
What is,
He can get an extra lunch every day. Barbara, this is your chance.
So what we can do to help you
Yeah.
by and large is get our friends and family
To upload it to GEDmatch and defamatory DNA, yeah. That would be cool.
Absolutely.
done.
Love it.
Yeah?
I have a question. That it's probably been at least 10, 15 years ago. National Geographic did a big DNA survey, right? They were capturing all kinds of stuff. And their objective was they were trying to show migratory patterns, but they were also trying to tie everyone back to a common Eve and a common Adam. And as far as I know, they made it back to a common Eve. Do you know, did they ever make it back to one man?
I don't think so.
Okay.
And I'm not even sure that it's really totally feasible. I mean, we're starting to see that that not only that there were Neanderthals and Denisovans, but there are other hominids that were also around at the same time. So I think we're learning a lot more about what was going on. So I think it's a little bit unrealistic probably to go back to a single person.
Yeah, our family did the same thing with National Geographic, and they shut down their system.
Right.
When they did that, I was wondering why. Are they worried about the liability of the personal information, or,
I don't actually, and there was a very strange interview. Oh gosh, I'm so bad with names. I'm not going to remember the name of the guy who was actually heading up that project. And he made some very, very strange comments. And so, you saw them? Do you remember what his name was?
I don't remember his name, but all about the privacy concerns that I just mentioned, those, like, you just started to wonder, wait, where is this stuff?
Mm-hmm.
Where is it going, and how are you using it? Yeah.
Yes, I think it's a little unrealistic to even think about your privacy in your DNA, because if you think about it, you saw the numbers, you're sharing DNA with all those third and fourth and fifth cousins. And,
It's already too late.
It's already too late. It's exactly right, yeah.
Plus all the DNA we've all given off just by sitting here.
Exactly, yeah. Yeah, we're shedding hair, we're shedding skin cells, yeah.
So if we can just, like, trim hair, give it to you.
Well, that begs the question, is there a service that you would recommend to have our DNA run so
Yeah,
that we
I'd love
can
to do that. Yeah. I'd love to do that.
What's the best
For
one?
those
Well, the only two that we can use for the forensic samples right now are GEDmatch and family tree DNA. There's another company called Othram they've also set up a database, and they do whole genome sequencing, and they actually probably have the record for solving a case with the least DNA. I think they got down to something like 12 picograms of DNA, which is basically touch DNA.
What is a picogram?
Well, we're for most of this stuff, we're talking about nanograms, so that's 10 to the minus 9. So a picogram is 10 to the minus 12 grams.
He asked, he wanted to know.
It's not very much DNA. It's maybe, 2 or 3 cells.
So it might be what you would leave behind after you touch something.
Yeah.
That's big then.
Which actually is the next big thing, is touch DNA.
So let's get back to, you're saying there's an organization that we could give our DNA to that would sequence our DNA that then could be put into these
I think that, well, no, they're actually asking you to upload to their database. So what you've already done, it's,
I'm trying to get back further than that, because National Geographic has completely abandoned my family's DNA. where do I get my DNA sequence so that I can then upload it to the database?
Well, you can, if you want to do whole genome sequencing, there are a number of companies that will do whole genome sequencing.
23andMe is for the account.
Ancestry.
Okay, gotcha, thank you.
And there are some other companies. So Ancestry and 23andMe are doing the SNP profiles. Family Tree DNA is now doing whole genome sequencing on, at least on forensic samples. I don't know if they do them on, if you went into and asked them if they would do it, but there are some other companies. There's a company called, I think it's Full Genomes or something like that. For around $1,200, they'll do whole genome sequencing. So then you've got the whole ball of wax. It's a very, very big file. But you could do it there.
You know, we're talking about the DNA, but if you were just trying to figure out your own genealogy, you probably don't even really need to do that, right? I mean, you can get all of your genealogy for free on the internet.
Well, you might want to do it in that you're getting every single piece of information that you could possibly ever want. And so if there was a change in what was being used by, for example, the direct-to-consumer companies, you wouldn't ever need to retest because you've done it all. So you'd get all of your mitochondrial stuff, you'd get all the haplogroup stuff, you get all the STRs for doing your Y-DNA. So you're getting a lot of information if you particularly wanted to, be pursuing some esoteric relative.
But if you're trying to go back 5, 10 generations of your own family, you can get that
Yeah.
without testing, right? You just go on the internet, that's all there today for free.
Mm-hmm.
Won't help you solve a crime, that it will help you find people.
For some people, yes. For some other people, no.
5 generations, pretty much.
I had a family who came across on Ellis Island and have entirely new names, things like that. So it can be difficult to translate that.
I'm saying 5 generations, meaning 5, like your parents, then your grandparents, great-grandparents, even Ellis Island. My family came to Ellis Island. And name change. In fact, I was showing that to Barbara last night. We saw 3 spelling changes as they came from Scotland.
But it does depend also, though, on your racial background, because the folks who are in the databases, the reason that we're arresting a lot of old white guys is because
It's retribution.
the people who are right. So the problem is that most of the people who are in the database, 80% are white, or they're Northern Western European ancestry. I've been working on a number of these are sexual assault kits out of Cuyahoga County in Ohio, and so we've got 20 cases that we're working on. Of those, only 3 are white. The rest are all non-white, and they are much more difficult to work on because where you test, you've probably got 3,000 to 5,000 matches in family tree DNA and GEDmatch. For somebody who is black or Hispanic, you might have 1/10 of that. So your odds of getting that second cousin match, of getting matches that you can connect and get your most recent common ancestor to go down. And so it's much, much more difficult to work on those cases. I'm sorry, excuse me?
I can't commit crime.
No, we'll find you.
You just won't get caught.
So yeah, it's really a numbers game. And so what we've started doing for some of them, particularly for the unidentified remains of their children, there's a national center that does a lot of that, the National Center for Missing and Exploited Children. And so what we started doing is when they put up their NamUs listing on the NamUs website for missing people, is putting in there that we know that this person is whatever background. Please, if you're from that part of the country, you're that racial background, please, if you've tested, upload to GEDmatch. We have a case that we're working on. It was a head found in a backpack up in Basto, California, which is up in the high desert, which is basically a dumping ground for all kinds of stuff. And basically the person was scalped. And obviously there's no hands, there's no nothing. We do have the racial background, she's Hispanic. And so not very many matches in the database. So we're really appealing to the Hispanic community, particularly if they're from Southern California. Please, consider, if you've tested, uploading, or if you haven't tested, consider testing. And we have some other cases where we've got somebody who's non-white, again, looking at, if they're from a small community, seeing if we can maybe get people to, well, we have one, this is out of Opelika in Alabama. It's a small black town. The detective is black, the victim is black. And so what Family Tree DNA is doing is they're actually donating some kits. And they're going to go around to the old folks' homes and see if they can get some of the elderly residents to test so we can get more people into the database. So there are some things like that we can do.
Yeah, I was going to ask if there was like a donation arm of some of the testing places where they help get more people.
Well, thank you for bringing that up, because actually we have just incorporated as a nonprofit, and once we get our IRS nonprofit status, we're going to actually start soliciting donations, so that if it's a small town police station which maybe doesn't have funding to do a lot of stuff, that we can actually try to fund it for them. Typically, you're looking at, for doing the testing on forensic samples plus the genetic genealogy, you're probably looking at between $7,000 and $10,000. So we're actually going to start doing that. We've actually opened a page on Facebook. It's called Firebird Forensics Inc. And at some point we're going to get it set up, as I said, once we have our IRS letter saying that we're an approved not-for-profit, then we will be soliciting donations.
It's very interesting. So you could just go into a community where a murder occurred and then just start to say who's willing to donate, and probably lots of people will.
And actually, Othram does that. That's the company that I mentioned that does the whole genome sequencing, and it also has their own database where they're trying to get people to upload. They're actually doing, they do crowdsource funding, but they're not a not-for-profit, and so, but they do solicit donations to pay for, everybody's favorite cold case. And there are all kinds of folks out there who really do follow a lot of these cases, and they'll send in the name of a case that they would really like, work to be done on, and they might help fund it.
Sounds like a great true crime tag.
Yeah.
It's my marketing brand of hey, if you live in this community, upload, because you say, if you live in this community, recognize this face, call us. But in this case, it's, if you live in this community, please upload your thing so we can help solve this crime.
Mm-hmm. In fact, we just solved one 2 days ago. It was actually another hair case. We're just waiting on swabbing the sister. This was an unidentified human remains case. But it's one of the ones that the true crime followers have posted on Othram. They really want somebody to work on this case. So yeah, we've now just solved it. So.
How many people can do this hair thing now? You had the guy that's in California. Is there more now?
Supposedly, Bodie, and I think Bodie's in Virginia. They're a big-time forensics company, been around for a long time. They supposedly are now doing hair. I have no idea how accurate it is. As you can see, it's a little bit tricky, and working with it is a little tricky. You have to really look carefully at what you have.
Well, that was something that you had said about posting a new file or posting a new.
Mm-hmm. Yeah, well, we actually, right.
You're adjusting the algorithm to fill in the gaps.
Right.
So that then you can get.
Yeah, we make sure that we're getting matches that are consistent with what we should be getting. And of course, with the Allenstown people, it was easy because we had the parent-child situations. With the one with Precious Jane Doe, it was a little more difficult. We didn't have that kind of control there. So it was a case of going back and saying, okay, is this making sense? Were these people really the right relationship? And it turned out they were, yeah. But we had to adjust the numbers because the numbers we were getting, because of all the little tiny pieces were too high. And so we were going back and recalculating them, just using the bigger matches, or the bigger segments.
I find that part fascinating.
It's fun.
It is. What you're how my brain works.
Mm-hmm.
And so like I'm just like, oh, I get it. I get it.
Yeah, and you learn all kinds of interesting stuff about your own family when you do your own stuff. Yeah.
Yeah. Have you done your?
Oh yeah, I've even done DNA testing on my cats.
Their Egyptian ancestors.
Well, it's kind of interesting. I just one of my cats unfortunately died, and I just got some kittens, and so I've just done them. And one of them, so they're linimmates, the two that I got, and so one looks like a snowshoe Siamese, and the other one is a little tabby. And when you pick her up, she's this floppy thing, like she's got no bones in her. And so I just got her results back a couple of weeks ago, and she's got a fairly large amount of ragdoll in her. Well, of course, the ragdoll, that's exactly what they are. They're this floppy cat. So yeah, it's really kind of fun. Alrighty, anybody, do you have any other questions? Okay, well, I hope you enjoyed.